So good evening all, this is Sanjay from Madras Medical College doing second year MBBS. Today we are going to discuss about necrosis in detail. So necrosis is a pathological process that is the consequence of severe injury. So the cause of necrosis are ischemia, as we discussed earlier, ischemia, loss of blood supply, which leads to loss of oxygen to tissues and exposure to microbial toxins, burns, another cause of physical and chemical injury. And necrosis is characterized by denaturation of cellular proteins and leakage of cellular contents to damaged membranes, local inflammation, and enzymatic digestion is very important. Okay, because cell membrane is also damaged, cellular proteins are also damaged, denatured, and it causes local inflammation and enzymatic digestion. Next, we will see about other facts about necrosis. So the cellular contents will leak out of the cell through damaged plasma membrane we have seen earlier into the extracellular space. Okay, so this point is also very important. As a result, the cellular contents also leak to extracellular space where they elicit a host reaction, inflammation. So this is what how inflammation is formed. So inflammation is formed due to leakage of cellular contents to damaged plasma membrane to extracellular spaces where they form a host reaction or inflammation. So there are some specific substances released from injured cells which are called as damaged associated molecular pattern. Damage associated molecular patterns. Okay, these are specific substances that are released from injured cells, and this include ATP. That is, ATP means these also damage associated molecular patterns. It is released from mitochondria which has undergone damage, and uric acid is caused due to breakdown product of DNA, and numerous other molecules that are normally confined within healthy cells. Okay, so next we are going to see about receptors, how these molecules are recognized. So these molecules are recognized by receptors present in macrophages, and they trigger phagocytosis. What is damage associated molecular patterns means these specific substances will be released by the injured cells. Okay, and what will happen is these molecules will bind to the receptors present in macrophages and stimulate phagocytosis as well as the production of cytokines. So two functions are there: triggering of phagocytosis and production of cytokines that will produce inflammation. So inflammatory cells, as you all know, will produce more proteolytic enzymes. Okay, so necrosis, as I seen earlier, the same point is repeated here, and then for example, the intracellular proteins present in serum blood is the basis of detecting the defect of tissue specific cellular injury, because if we take example cardiac muscle cells, they express specific protein, cardiac protein, contracted protein called troponin. Bile duct epithelium will expresses alkaline phosphatase, an isoform of enzyme alkaline phosphatase, bile duct, and hepatocytes will express transaminases. That is, these products are released in these specific body regions due to damage to cell membrane caused by necrosis. So necrosis, as you said earlier, will cause loss of membrane integrity, and increased serum level of these protein is found. Okay, and biomarkers that are used clinically to quantify tissue damage. So 2 hours after myocardial cell necrosis, the cardiac specific troponins can be detected. Okay, after 2 hours, we need to wait for 2 hours. Only then we can find cardiac specific troponins present in the blood. So because of their sensitivity and specificity, the serial measurement of serum cardiac. So the measurement of serum cardiac troponin has a central role in diagnosis. You can diagnose easily whether the patient is suffering from myocardial infarction or not. Next, I will be discussing about types of necrosis. So necrotic cells will show increased eosinophilia and hematoxin stain because loss of cytoplasmic RNA and other nuclear genetic material, and due to the accumulation of denatured cytoplasmic proteins or eosinophilic, will cause increased eosinophilia in necrotic cells. Okay, that is important. And When enzymes have digested the cells organelles, the cytoplasm will become vacuolated. Vacuoles are formed and dead cells is replaced by myelin figures, which is a whole phospholipid precipitase. This is myelin figures. These myelin figures are either phagocytosed by other cells or further degraded into fatty acids. Okay. So electron microscopy can see that necrotic cells are characterized by discontinuous in plasma and organelle membranes and marked dilation of mitochondria with the presence of large amorphous densities. Next we'll see about nuclear changes. So in the case of nuclear changes, uh, due to the breakdown of DNA, like one of three patterns will be a nuclear change. That is, first one is karyolysis. So karyolysis is nothing but the base of failure of the chromatin will get faded. That is very important. That is karyolysis. It is a change which reflects loss of DNA because of enzymatic degradation by endonuclease. So in other words, we can tell like karyolysis. Karyo means nucleus, lysis means degrading. So degradation of DNA by endonucleases is nothing but karyolysis. And here the base of failure of the chromatin will fade. And pycnosis. Okay, it is characterized by nuclear shrinkage and increased basophilia. So in pycnosis, there is a nuclear shrinkage and increased basophilia because in pycnosis the chromatin condenses into a dense shrunken basophilic mass. See here, I am here, pycnosis, shrinkage of nucleus and increased basophilia. Whereas in karyolysis the basophilia will fade, but here there is increased basophilia. Here the chromatin gets condensed into a dense shrunken basophilic mass. And in karyohexis, the pycnotic nucleus undergoes fragmentation. So that is what all about karyohexis. That is nuclear changes in 3 nuclear changes which occurs in necrosis. Next, I am discussing about types of necrosis in detail. So if we take coagulative necrosis, it is a form of necrosis in which the architecture of dead tissue is preserved for a span of at least some days. How it occurs, we will see it in detail. See here, if the genital injury will denature not only the structural proteins present but also the degradative enzyme. So even degradative enzyme is also destroyed. So it blocks the proteolysis of the dead cells, and as a result, intensely eosinophilic cells with indistinct nuclei or reddish nuclei can be seen for days or weeks. And after that, finally, necrotic cells will broken down by the action of lysosomal enzymes, and also they remove the debris of the dead cells by phagocytosis. Okay, so for example, ischemia caused by obstruction in a vessel may lead to coagulative necrosis of the supply tissue in all organs except brain. And what is an infarct? This also may be an MCQ question. This is a localized area of coagulative necrosis that is called as infarct. Next, you will go to liquefaction necrosis. As the name indicates, it is opposite to coagulative necrosis. See, digestion of dead cells is the main feature of liquefaction necrosis, resulting in transformation of the tissue into a viscous fluid. So it is seen in fungal infections, focal bacterial infections, and other microbes which stimulate the accumulation. What I am highlighting is very important: accumulation of leukocytes and the liberation of enzymes from the cells. And the necrotic material found in the liquefactive necrosis is creamy yellow, frequently creamy yellow, and it is due to the presence of leukocytes that is called pus. Okay. Okay, so next, for example, hypoxic retrocellular in CNS often manifests as liquefactive necrosis. Next, we will see gangrenous necrosis. So it is not a specific pattern of cell death, but it is a term which is commonly used in clinical practices. See here, if you take our leg or lower limb that has lost its blood supply and has undergone necrosis, usually coagulative necrosis occurs. But here, when bacterial infection is also superimposed, there is more liquefactive necrosis because the bacteria will produce the degradative enzymes and which could cause the action to occur, and the attracted leukocytes will give rise to wet gangrene. Next, let's see about caseous necrosis. In caseous necrosis, it is encountered in foci of someone encountered in tuberculous infection. Okay, so caseous is nothing but it is a cheese-like, that is it will be like a friable white appearance. The part of the necrotic tissue will be like friable white appearance, and in microscopic examination, the necrotic area appears as a structureless collection of fragmented live cells and amorphous granular debris. Okay, so this appearance is a characteristic of focus of inflammation. So what it reveals a friable white appearance of the area of necrosis, structured and consists of structureless collection of fragmented or lyse cells and amorphous granular debris enclosed in distinctive inflammatory border. Okay, that is caseous necrosis. Next, let us see about fat necrosis. See here, in fat necrosis, it typically results from the release of lipases especially from pancreas, pancreatic lipases into a substance of pancreas. So pancreas releases substance of pancreas and peritoneal cavity. So this in acute pancreatitis, the leakage of pancreatic lipases, activated pancreatic lipases occurs, and in this disorder, the pancreatic enzymes will liquefy membrane of fat cells. First, liquefaction of membrane will occur, and triglyceride esters are split by pancreatic lipases. What is fat saponification? May be asked in short note. It's nothing but fatty acids which are generated combined with calcium with glossy visible chalky white areas called as fat saponification. So that's it. Next we see fibrinoid necrosis. So fibrinoid necrosis is a special form of vascular damage. Here it is involving the blood vessels. It is seen in immune reactions involving blood vessels, and it occurs when antigen complexes and antibodies are deposited. That is very important. When it occurs when the complex antigen and antibodies are deposited in the wall of arteries. Okay, so what is fibrinoid? It means fibrin-like. That means the deposits of these immune complexes. So immune complexes will be deposited, as we seen earlier, antigen antibody complex, and together with plasma proteins released out of vessels will result in a color that is bright pink and amorphous appearance in hematoxylin and eosin stain, which is called as fibrinoid or fibrin-like by pathologists. So that's it. So lastly, we have seen about necrotic fibrosis and fibrinoid necrosis, not necrotic fibrosis. Fibrinoid necrosis, fat necrosis, we have seen. So it's very easy topic. Okay, next. So necrosis is a pathological process in which cellular membranes are destroyed. This portion is very important. So reversible cell we discussed earlier. Plasma membrane blebbing will occur, and then loss of microvilli and mitochondrial swelling and dilation of endoplasmic reticulum and eosinophilia. Whereas if we take patents, it is of various types: coagulative, liquefactive, gangrenous, caseous, fat, and fibrinoid. So blebbing means it is a process. So here often we hear the word blebbing, especially in reversible cell injury. What happens is the cell's plasma membrane will bulge out in a spherical shape. That is what bleb. See in this diagram you can see blebs clearly. See, these are the blebs. See, it is among these spherical areas. Spherical areas, here blebs are seen. So that's it guys today. Next we'll be dealing with apoptosis, I guess. Next topic is apoptosis. We'll be dealing with apoptosis in detail in next video. Thank you.